Good afternoon. I am Professor Sir Mark Caulfield, and I'm going to speak to you today about transforming genomic healthcare. I'm Chief Executive of Barts Life Sciences. I'm going to talk to you about the 100,000 Genomes Project, which was the brainchild of a former Prime Minister of the United Kingdom, David Cameron. This is reading as much as we can today of your and my genetic code, the 3.3 billion letters that make us the individual we are, but can also sadly carry susceptibility to disease. This program was focused on whole genomes because it was felt at the time that these would be tractable to healthcare, particularly in rare disease, infection, and cancer. And over 97,000 people from families with rare disease or with cancer or infection and their relatives participated in the program. It's now up to over 40 petabytes of data collected, and we had to change the infrastructure in the NHS and create 13 genomic medicine centres, which have now become 7 genomic medicine service alliances encompassing 200 hospitals. At the peak of the program, 5,000 frontline NHS staff touched this project at some point during their working week. This is important because it creates the champions who will support the adoption of this legacy in the health system. And we invited researchers from around the world, from bona fide institutes, to join us to drive up the value of these data, and over 3,000 from 33 countries have joined. I'm going to focus a little bit on rare disease. Rare disease is 6% of Western populations. Around 1,200 disorders with unmet need were addressed in this program. These were all volunteered by clinicians from the health system, health professionals, or academics or industry. They had standardized criteria for entry and standardized data collection using the human phenotyping ontology, which means my data, if you use the same ontology, can talk to yours. We automated analytics, which didn't exist at the time, and we involved the NHS in the selection of gene panels and the closure of cases. Along the bottom here, you can see on the range of numbers of families by different system that we worked on with different rare diseases. I'm going to share with you some diagnoses from the project. And first of those is a very sad case of a 4-month-old with fluctuating neurology, repeated infections, and who was admitted to the neonatal intensive care unit at birth and died sadly after 4 months in that same intensive care unit. And died with no diagnosis, immune testing had been negative. His parents were devastated, and they asked to enroll the deceased child's DNA in the program so they could find out whether or not this would inform a future reproductive choice. Then mum became unexpectedly pregnant and said, I don't want to know the diagnosis. We found a pathogenic change in the transcobalamin 2 gene, which transports vitamin B12, something you and I have eaten today, inside cells. His brother was born, and because we knew where to look, was tested within 1 week, and sadly was also found to be affected. But because of case reports that high-dose vitamin B12 had ameliorated the symptoms, he was given this, and this child has had a completely different course and is alive today. Second case, a 10-year-old girl admitted to ITU with repeated severe life-threatening infections. These repeated admissions cost the NHS £356,000. Detailed immune testing had made no diagnosis, and mutations in CTP synthase 1 gene affecting B and T lymphocyte responses to infection had dampened her ability to respond appropriately. This was cured with a bone marrow transplant, and her siblings were tested and were not at risk of these infections. The final case is where the genome reprofiles a diagnosis. This is a 5-year-old boy with unexplained anemia, developmental delay, short stature, and constipation. He had a diagnosis given to him clinically of Diamond-Blackfan anemia, which is a limited lifespan due to cancer risk. Because his clinicians had told us that he had developmental delay, we looked at intellectual disability genes and found that he had a pathogenic thyroid hormone receptor alpha mutation, which meant that his thyroid hormone receptor could not respond appropriately to thyroxine, an everyday hormone that you and I use to manage our metabolism. He's now receiving a high dose of thyroid hormone replacement, and he's a transformed child. You can see his video on our website. So what I'm sharing with you here is the average diagnostic yield of about 25%. across the first 2,183 families. This will be published in the New England Journal on the 7th of October. And you can see the impact of different family structures. So a single family member that's affected, you get about a 17–18% diagnostic yield. It increases dramatically with one parent and further with two parents. And that shows the impact of family structure. So if you do research on the genomes and it's a likely single gene disorder, which often rare diseases are, then you can have a 37% diagnostic yield, but somewhat less with more complex etiologies, such as 12%. And in the pie chart on the right-hand side of your slide, you can see in the blue the things that you might have found with pre-existing tests in panels, and in the yellow, green, and orange, what you can now find if you add a whole genome. Here is the diagnostic yield across those 2,183 families, and you can see that's 25%, roughly at the threshold above 25%, where there are a number of disorders that achieve appreciably greater diagnostic yield. And bear in mind that none of these people had a diagnosis before they entered the program, and this is addressing unmet diagnostic yield. We've used this information to inform clinical care, and there's now genomic testing live in the NHS with whole genomes, and I'll come back to that in a moment. About 24% of the diagnoses had an immediate clinical utility. They changed care in several different ways, which you see there. You became eligible for trials, you had additional surveillance, you could inform reproductive choice, there was a medication you could have, or you could have a dietary shift or vitamins to ameliorate your disorder. Importantly, it's worth looking at the Diagnostic Odyssey and what that costs the health system. So these children spent a median of 68 appointments, that's 6 years not getting a diagnosis, whereas unaffected relatives spent 18 months and over 18 appointments over 120 months. Post-diagnosis, there were fewer focused clinical episodes, showing that the diagnosis hones the clinical care. These affected participants used 183,273 hospital episodes of care and cost 87 million to the taxpayer, compared to their unaffected siblings who had 53,000 episodes of care and cost 21 million. So this is a real cost. And if you now think that the genome is less than $800, you can begin to see how this technology could penetrate the health system. I'm going to move to cancer, which proved to be very challenging. Cancer is essentially a disease of disordered genomes. We reached 17,339 cancer participants. This is the largest such cancer resource in the world. But we found that we couldn't make formalin fixation, the standard preservative for tumor tissue, work. So we re-engineered 400 molecular pathways and brought live fresh tissue whole genome sequencing in the NHS. And this means we have very high quality genomes. And you can see here one of the cases who had a clear change in care. So this is a family tree. The squares are men, the circles are women. If you've got a diagonal line, you're sadly deceased. And if you're shaded, you're affected. So the shaded green circle is a lady who had breast cancer, no family history of breast cancer, would never have been tested for BRCA mutations, but she had a BRCA2 mutation in her tumor and in her germline. And she transmitted that to her daughter, who then had a predictive test and now can actually have intensive breast screening, consider risk-reducing surgery or chemo prevention. But the lady herself got to be in the Olympia trial and received the Laparib, and her male family members can also consider BRCA2 testing because they may be at increased risk of prostate cancer. So this illustrates in one family the impact family-wide of these findings. So here's another slightly technical slide, but let me take you through it. So one of the cases that we have had a brain deposit of a tumor. This was a secondary tumor, so it wasn't the primary, but we couldn't tell from the histology what the primary of the tumor was, and this helps with therapy. So we found some mutations in the tumor, which suggested this could be potentially a lung cancer, but we went on then to look at tumor mutational burden. This is the number of mutations per million bases of the genome, and we found that this person had a high tumor mutational burden in the range of colorectal, lung, or melanoma. And the detective story becomes more focused, and we catch the cause when we discover that there's a smoking signature in the deposit, which makes it highly likely that this is lung cancer. So we've taken a cancer of unknown primary where we didn't necessarily know how to treat the patient, and we've converted it to something where we have some sense of what they may respond to. Here's some data showing a survival curve. And so up the side, you've got percent survival. So the further down the curve is, the less people alive. The pink curve is PIK3CA, a known mutation, a known genotype that predicts survival, and the mutant. And you can see that the mutant survives better than the wild type. This is a known association, but we've been able to demonstrate this in our patients after 4 years because we have longitudinal life course data. So everyone's followed up for their entire lifetime. So this is a known finding, and the reassurance is we've been able to duplicate this. But this also means not only can we find new signatures of harm and bad outcomes, but we can also find late signatures of therapeutic influence and potential adverse outcomes from therapy, which goes way beyond the length of time the trials have followed the patients. Here's what the clinic did with the data. So of 3,244 patients in the West Midlands Genomic Medicine Centre, 781, 25% were returned for reevaluation by the Genomic Tumour Board, and of those, 477 had a new action. 53 had something called DPYD, which is a genetic variant which changes your activation of 5-fluorouracil and capecitabine, drugs used to treat cancer. And this can cause severe organ failure or real problems. So 53 people were able to have a reduced dose or avoid those drugs altogether. 21 people had a different licensed therapy that they would have had because of the tumor signature. And some 11 had unlicensed therapy, and 204 took part in a trial. 43 had high tumor mutational burden, so these would signify potential use of immunotherapy. 69 were referred to clinical genetics, and 76 had no specific recommendations. So you can see that this is not a useless result. It can be used to influence cancer care. Just to say, we've looked at over 76,000 people's genomes now, and we find multiple variants that influence drug handling. I've spoken about DPYD, but you can see that 30% of us, which was known before, but we can identify it with whole genomes, have a risk genotype, which means we may not activate clopidogrel. And in 100,000 genomes across 76,000 people, 99.5% of us possess a clinical pharmacogenetics international consortium actional gene drug pair, and 25% of us may have as many as 4. So we don't entirely know what to do with this data. And so we needed a coalition of research intellects, and we openly advertised, and 3,580 researchers from 33 countries volunteered. And they've won 50 million in grants for themselves, a 43% success rate. And this has allowed them to join us inside our data center in the UK, where you can work on 4 billion clinical data points and 111,000 whole genomes. We've used this data to transform our NHS and create a new NHS Genomic Medicine Service. And that involves national genomic testing through 7 NHS genomic laboratory hubs. And they do testing that's not whole genomes, every other test. And we also created a national test directory. It's a directory, not a catalogue or a guideline. We reprofiled 300,000 tests, upgraded 25% to new technologies. Everybody is offered consent for research using a single national unified genomic consent, which allows us to longitudinally life course follow the patients, recall them for research, and make the data available from a secure data center in the UK to researchers worldwide in de-identified format. And thereby we're creating a national genomic research library. Genomics England will continue to run the whole genome sequencing, and the NHS has also created 7 genomic medicine service alliances. I'd share a little bit of information about COVID-19. We've done a study which is on MedXRIV and in review at Nature on 7,491 severely ill people with COVID-19 from 224 UK intensive care units. Compared those with 48,400 unrelated controls, and we validated those findings in the Host Genetics Initiative, and also looked at transcription changes and looked at rare variants. We've discovered 25 signals that are genome-wide significant, 22 fully replicated, 15 when brand new, 3 signals appear to be ancestry specific in South Asians, and transcriptome-wide analysis has shown multiple signals with significant exchange in the expression of genes. We haven't found anything with rare variants at the moment, but we're still a work in progress, and there are 2 putative therapies. This is something called a Manhattan plot, where the chromosomes are arranged from left to right, 1 to X, and the log p-value is up the side. The red serrated line means genome-wide significance, so you can see the different gene regions, and these highlight multiple genes involved in innate immunity, inflammatory pathways, and the ABO secretor blood group. We've placed participants at the heart of our program and throughout it. They shape our strategy, they sit on our committees, and we've also done research with the public and patients around key public behaviours in relation to the NHS social contract. That's a free at point of care system of healthcare. And this public consultation revealed the public think that we should, as members of the community, exhibit reciprocity. We should be reliable in turning up to our appointments, try and support the NHS. We should be altruistic. We should allow our data to be used for research, and we should show solidarity. We should accept progressive taxation and treat all with equality and respect. But they have some red lines. They're not ready yet for gene editing of their genome. At the moment, we are looking at gene editing in terms of particular tissues and cell groups, but not the whole organism. They also recognize the need for commercial access to the data to develop therapies and diagnostics, and they're comfortable with that, but they're not keen on this being used for marketing, and they don't want their data used for a surveillance society. So we now have a new program which is funded for 1 year, and we're looking for funding for a further 3 years. This will focus on diversity in genomics because there aren't enough diverse people involved in genomic studies. Also cancer, where we'll be looking at multimodal cancer care, and the newborns, where 1 in 190 live births in England could have 1 of 600 conditions. That means it could be treatable by a mineral, a vitamin, a dietary change, and only 8% are expensive. And we're going to look in the first 5 years of life where we could detect people where an intervention exists that could reduce harm or avoid it altogether. We've also consulted the public and patients, and in the green on the left, you see what the public's views were about the program in terms of what we should do. They were excited by the pace of technological evolution. They felt it should be a paradigm shift, and they also felt that it should be equitable and that it should include all sectors of society. In the blue, what you can see is that it must involve a representative section of the UK, needs to be in lots of health settings, not just the ivory towers, needs to robustly test the evidence and use transparent processes. So to summarize, the National Health Service in England now has a National Genomic Medicine Service providing consistent, equitable care for 56 million people, operating to national standards, specifications and protocols, a single approach to standardized genomic consent, a national test directory which covers all testing from single gene to WGS, and a single UK genomic research library, which will be available to researchers worldwide in de-identified format. And the future is a global coalition of intellects, so I look forward to seeing you working on the data. And I should like to thank everybody who made this possible, the United Kingdom government who championed this for 8 years, and also all the people in the NHS and our universities and our funders who worked on this, and the most important people in the program, who are, of course, these people. They are our participants, and they really crave an answer for why they're like they are, and they're hoping you will come and help find it. Thank you so much for having me here today. It's been a real pleasure. Thank you.